Good afternoon friends and welcome to EduSite live lectures. Dear friends, today's topic for discussion is Molecular Biology, Genome Organization Part 2. To discuss this topic, we have with us our subject expert Dr. Charu Dogra Rawat. Dr. Rawat here is an assistant professor in Department of Zoology in Ramjas College, University of Delhi. Without further ado, I would like to welcome to our studios and request her to start a lecture. Welcome ma'am.
Thank you. Good afternoon everyone. So today I continue with the Genome Organization, and I talked about prokaryotic genome organization in my last lecture, and I'll continue with the eukaryotic genome organization. So we have done the structure of DNA, how it looks like, but now we are doing how it is packaged inside the cell. So we'll talk about the genome organization in eukaryotes today. So the learning objectives for today's lecture will be that you will understand at the end of this lecture the building blocks of chromosomes, which are called as the nucleosomes. You will have the understanding of the structure, the function and the positioning of these nucleosomes. You will also understand the chromatin organization at interphase. I started with this a little bit in my last lecture, but I'll continue with this and finish it this time, that how the chromatin is organized at the interphase division. When the division of the cell starts, you will understand the chromatin, or basically the chromosome organization at the metaphase stage of the cell cycle, and you will also understand the structure and function of different parts of a different components of a metaphase chromosome. So as we talked about that the chromosome structure changes as the eukaryotic cells divide. So when the eukaryotic cell is dividing, it goes from an interphase and then follows mitosis or meiosis, whatever the division, the kind of division that is occurring. But as we mentioned, there are stages in mitosis and meiosis which constitutes the prophase, the metaphase, the anaphase and the telophase. So as it passes through all these stages, the chromatin or the genome inside the nucleus changes. In the interphase, the chromatin is quite diffused, but at the end, say for example in metaphase and anaphase, when finally the replicated chromosomes divide and they segregate into two daughter nuclei, the chromosome is much more condensed. So we will follow the genomic organization throughout these phases and stages, basically the two stages, the interphase and the metaphase stage, through my today's lecture. So as you can see, the chromatin is organized, or the levels of DNA packing can be seen. The first is basically the double DNA helix, just a DNA helical structure, which is approximately 2 nanometers in width. Then as you go along, then there is a structure which is basically the nucleosomal arrangement that appears as a beads on a string. So we will talk about in detail about all these structures. That gives the width of a 10 nanometers, and that gives a 6-fold compaction. Then there is this nucleosomal string, the beads on the string is is basically swirled around itself, and therefore that gives rise to the 30 nanometer chromatin fiber of the packed nucleosomes. The further up is that these particular packed nucleosomes are then thrown into loops, giving rise to a 300 nanometer structure. Then these loops are in the metaphase, present in the metaphase chromosomes, attaining higher order of compaction. And as you can see, the metaphase chromosomes look quite different from how the double helical structure of DNA was there. So the maximum compaction that is attained is around 1000 to 10,000 folds of compaction can be attained when the metaphase chromosomes are there. So we will follow this chromatin organization or the level of DNA packing, and we will start with the nucleosomal, the 10 nanometer and the 30 nanometer fibers, basically the nucleosomal organization in the eukaryotic chromosomes. For the nucleosomes, as I said, the chromatin is basically the DNA associated with the proteins. The major proteins of chromatin are the histones. They are small positively charged proteins containing a high proportion of basic amino acids such as arginine and lysine that facilitated the binding to the negatively charged DNA molecule. So these are the major proteins of the chromatin, the histones protein, and they get together to form what is called as the nucleosome, around which the DNA is wound. So these histones, there are 5 major types of histones. They are called as H1, H2A, H2B, H3 and H4, and as you can see the molecular weights are different. The H1 is higher, it's a heavier protein. It is called as the linker histone protein, and then there is a core histone proteins which are H2A, H2B, H3 and H4, which are approximately of same molecular weights ranging from 11 to 13 kilodaltons. And then you can see the number of amino acids which is present, and obviously the H1 has higher percentage of lysine and arginine which is there. In addition to the chromatin, they contain an approximately equal mass of a wide variety of non-histone chromosomal proteins. We will not go into detail about these non-histone chromosomal proteins today, but all I can tell you is that these non-histone chromosomal proteins are basically like the transcription machinery protein or the replication machinery protein, which get associated with the DNA to carry out different functions of the DNA which is there, such as replication or transcribing into mRNA, etc., or just for the regulation and expression of the genes. The regulatory proteins are also bound. So these are all non-histone components, but today we will talk about the structural aspect, which is basically the organization into nucleosomes. So histones form the major protein. So we will restrict ourselves to the histone proteins today. The core histones are H2A, H2B, H3 and H4, and they share a common structural fold. As you can see that there are alpha helices which are present. So there is an N-terminal chain and there is a C-terminal which is present, and in all the 4 core histone proteins, there are 3 alpha helices which are there, 3 alpha helical regions separated by 2 short unstructured loops mediate the formation of head to tail heterodimers. So these histone proteins or core histone proteins exist as dimers. So H2A forms a dimer and H3H4 forms a tetramer. So there are 4 protein molecules for H3 and H4 which joins together to form this H3H4 tetramer, and this histone fold is very important, which contains the 3 alpha helical regions separated by 2 short unstructured loops. The nucleosomes are said to be the building blocks of the chromosome. The basic structural unit of chromatin, the nucleosome, it was first described by the Roger Kornberg in 1974. So this is how pictorially a nucleosome would look like. So there is a histone core protein which is present, and then the DNA is swirled around this histone core. That DNA is called as a core DNA, and then there is a linker DNA which will make it join to the next nucleosome. So this entire portion with the histone core, with the core DNA and with the linker DNA is termed as one nucleosome which is there. Progressive digestion of nucleosomal DNA with micrococcal nuclease can tell us about the length of DNA which is swirled around the histone core. So if you take the DNA and you digest it with the enzyme which is a micrococcal nuclease, it will digest the naked DNA. It will digest the DNA which is not bound by the protein. So when you digest it, it will give you a 400 base pair and 200 base pair unit. So 200 base pair unit is a nucleosomal unit. So for example, it will cut at the points which are shown by the arrows, and it will create a 200 unit. Further, if we continue with this micrococcal digestion, it will cut the linker DNA, that 20 to 60 base pair part also, and when you run it, then you will get a smaller molecule which is the histone core molecule which is present. So when you run on the DNA, for example, if you are running it on an electrophoresis gel, the DNA molecules which will separate out will be in the units of 200 base pairs of the length, which is 200, 400, 600 and 800, as you can see on the right hand side agarose gel electrophoresis picture. So that basically tells us about the nucleosome, that it is around 200 base pairs in size. The nucleosome core particle corresponds to the beads visible by electron microscopy. So when you observe a DNA under an electron microscope, it appears like it is shown. The arrows, the beads are basically the nucleosomal, and you can see the spacing. If you cut it with micrococcal nuclease, it will digest the part which is not covered by the protein, and therefore it will give rise to a 200 base pair unit. an octameric protein core around which approximately 147 base pairs of DNA are wrapped in 1.65 turns of a left-handed superhelix. So, what is an NCP or what is a nucleosome core particle? It is an octameric protein core. So, there are 8 protein components, histone components, around which approximately 147 base pairs of DNA are wound, and they are wound in approximately 1.65 turns, and the superhelix is a left-handed superhelix. So, we know already from a DNA lecture that what is a left-handed superhelix. So, it is interwound in such a manner. The protein core itself is composed of 2 copies each, because there are 8 protein subunits. So, 2 copies each of the 4 core histone proteins, which are termed as H2A, 2B, 3 and 4. So, all these histones in 2 copies forms the octameric protein core, around which 147 base pairs of DNA are wrapped in 1.65 turns in a left-handed superhelix. So, that is the nucleosome core particle. Now, what kind of interaction occurs between the histone and DNA? We know that the DNA is a negatively charged molecule, and we know that histones are primarily positive charged molecules. So, now how do they interact, or what are the positions? We already know that DNA is a polynucleotide double helical chain. There is a major groove, there is a minor groove, there are bases which are stacked inside, and outside there is a phosphodiester bond. So, how does this histone interact with the DNA molecule? The massive distortion of DNA into a superhelix is brought about by the tight interaction between the rigid framework of the histone octamer and the DNA at 14 independent DNA binding sites. So, there are 14 independent DNA binding sites on the DNA by which it interacts with the nucleosomal core particle, like it is shown in the picture. The flat wedge-like surface of the histone octamer displays a continuous basic groove along the surface around which the negatively charged DNA is wound. Not a single direct contact with the base pair. So, the interaction between histone and DNA is primarily of non-covalent interaction. They are basically the hydrogen bonds which are present. Around 14 hydrogen bonds at these 14 sites are present between the DNA and the histone octamer. Of these 14 hydrogen bonds, the 7 hydrogen bonds are basically in the minor groove which are present. So, all the DNA interacts with the histone octamer at the minor grooves of the DNA itself. The interactions are made exclusively at the minor groove of the DNA, characterized by extensive hydrogen bonding, non-covalent bonding between the protein's main chain amide atoms and the phosphate oxygen atoms of the DNA backbone. So, it is a non-sequence specific kind of an interaction. The histone does not interact with the bases at all. It generally, it mostly interacts with the phosphate oxygen atoms of the phosphodiester backbone of the DNA. So, phosphodiester backbone of the DNA, therefore, it is not non-sequence specific interaction. 7 of the hydrogen bonds which are formed in the minor groove are there, but again it does not interact with the stable part or with the fixed part of the bases which is there. So, it is again non-sequence specific hydrogen bonding. It does not read the sequences. As we talked about when we were talking about the DNA structure, the major interaction with other proteins such as transcription machinery or regulatory machinery happens through the major groove of the DNA molecule. So, there they interact with the overall chemical environment and sequence specificity. The proteins come and interact with it. But the histone proteins interact through the minor groove, mainly associating themselves with the phosphodiester bond. Only association which occurs with the bases is again non-sequence specific, so they can interact in mostly all the minor groove, although it is a little biased as we will see the positioning of the nucleosomes on the DNA. But the major DNA interaction is non-covalent interaction, non-sequence specific interaction, and 14 hydrogen bonds approximately are formed between the histone octamer and the phosphodiester backbone of the DNA. So, that is how the histone DNA interaction takes place. Now, the nucleosome core particle assembly we can also talk about, because as we will see, this nucleosomal organization is a very dynamic organization. It is not fixed. So they keep forming and they keep again dissociating. For example, for replication, for exposing the parts of DNA which needs to interact with, say, transcriptional machinery. So this nucleosomal particle has to disrupt. So it will dissociate and immediately reassociate, for example, after replication. So there is a nucleosomal core particle assembly and disassembly which is taking place based upon the function the DNA is going to exhibit. So for that purpose, the organization of genome at this level is very, very dynamic. So the nucleosome core particle assembly, how does it occurs? In vivo, the chromatin assembly factors first deposit on H3, H4 tetramer on the DNA, to which the H2A, H2B dimer is escorted by histone chaperones. So this is very important because again the nucleosome organization is not spontaneous. It needs molecular chaperones. Chaperones are the protein molecules which aids in the folding of for a particular molecule, or which aids in the folding of protein molecules. So the histone chaperones, basically the first thing which occurs is the formation of the H3, H4 tetramer, and this tetramer gets associated with the DNA, and then with the aid of other molecular chaperones, the H2A, H2B dimer, two dimers of H2A, H2B will come and gets associated, and such a kind of a nucleosomal core particle with DNA wound around it will be formed. So this is how the nucleosome assembles, and when that part of DNA which is wound around the nucleosome is needed for any purpose, for transcription or for replication, then the disassembly of the components takes place. Also they just lie in the vicinity, and as soon as the replication is completed, they reassociate to form the histone proteins. Now one important aspect is that on replication the histones also are doubled, and then they are passed randomly onto the two daughter nuclei, because the new DNA which is formed needs to be associated with the same histones in a similar manner, but all are not formed from the old histones, or old histones are basically randomly distributed between the daughter duplexes which is formed, but the old is also retained and the new ones are also formed. So that is how the chromatin or the nucleosome core particle assembly takes place. There is another term which is used, which is called as the chromatosome. The extension of the DNA to 166 base pair length and the addition of the linker histone H1 gave rise to what is called as the chromatosome. So 147 base pairs of DNA wound around 4 histones, which is the 8 histone particles. The octamer core protein is the nucleosomal core particle. If you include the H1 linker histone and additional, say, approximately 20 to 60 base pairs of length of linker DNA, making it to 166 base pairs of total length, then that molecule is said to be a chromatosome which is there. And now it will have approximately 2 superhelical turns, because just in the nucleosomal core particle there is approximately 1.8 or precisely 1.67 superhelical turns which are there. So that term is called as the chromatosome. Now, so what is the role of this H1 protein, or what is the role of this H1 linker histone protein which we are talking about? It stabilizes the point at which the DNA enters and leaves the nucleosome core. So you can see that there is a nucleosome octamer core which is present, which is green in color, and the DNA is bound around it, and then the H1 histone, the C tail of H1, is stabilizing the DNA between the nucleosomal cores. So there is a spacing between the nucleosomes which is there, and this spacing is maintained by the H1 histone or linker histones of the H1 and H5 family, and therefore it stabilizes the point at which the DNA enters and leaves the nucleosomal core. So if we look at the packaging of the DNA with histone yields a chromatin fiber approximately 10 nanometer, the first level of organization for genome. So this double helical DNA is wound around the nucleosomal core particle and contains this nucleosomal linker histone and linker thing, and this packaging is approximately gives a fiber of 10 nanometer in diameter that is composed of chromatosomes separated by linker DNA segments averaging about 80 base pairs in length, and this packaging of DNA into such a 10 nanometer chromatin fiber shortens its length approximately 6-fold. So we need to package around 2 meter or 1 to 2 meter of eukaryotic chromosome or genome into a very small 10 raised to the power of -5 meter of nucleus. So this just gives the 6-fold, but that is the first level of organization which is there. So the packaging is basically 10 nanometer chromatin fiber. Now I will talk about that how these nucleosomes are positioned on the DNA. So two aspects I will talk about. The first aspect is that this positioning is done by bending the DNA double helix into 2 tight turns around the outside of the histone octamer, a process that requires substantial compression of the minor groove of the DNA helix. So we talked about this thing that now octamer protein is basically interacting with the DNA only at the minor grooves, and at the minor grooves this DNA bends down. Now bending of the DNA is easier when the AT base pairs are there rather than when the GC base pairs are there. So AT regions are preferred when they are present in the minor grooved inside, and the GC rich minor grooves are exposed towards the outside. So the nucleosomes are positioned. So it appears that the nucleosomes are very fixed on a DNA molecule. So the nucleosomes are AT rich sequences in the minor groove are easier to compress than GC rich sequences. So each histone octamer tends to position itself on the DNA so as to maximize AT rich minor grooves on the inside of the DNA coil. So this gives a kind of a specificity for the positioning of the nucleosomes or the DNA, or it gives rise to a kind of a fixed positions of the nucleosomes because it prefers the AT rich region, AT rich minor grooves with which the histone octamer interacts, and it puts the GC rich minor grooves towards the outside. So this is how the nucleosomes are positioned on the DNA double helix. We rise up to the next level, which is the 30 nanometer fibers. So what happens is that now this nucleosomal DNA thread, which is now nucleosome with DNA interwound around itself, is then twisted on its own axis, giving rise to the 30 nanometer fiber. So it is a higher ordered structure. The twisting is again a left-handed helix. The 6 nucleosomes are present per turn since it is twisting around its own axis. So 6 nucleosomes are present per turn. H1 and histone N-terminal tails stabilize this 30 nanometer fiber by interacting with adjacent nucleosomes. So this is very, very important. The tails of the histone proteins are very important. The N-terminal tails of the histone H2A, H2B, 3 and 4, and as well as of the H1, the linker histone basically stabilizes this 30 nanometer fiber structure by interacting with the adjacent nucleosomes which are present. Now, 2 models of 30 nanometer fibers are there. One is called as the solenoid model. The solenoid model is a model in which the linker DNA does not pass through the central axis of the superhelix, and the sites and entry-exit points of nucleosomes are inaccessible. So you can see in the diagram that the nucleosomes per turn are lying towards the outside, and there is no linker DNA which is found through the center of the helix which is formed. Another possibility is for the zigzag model, in which the linker DNA does frequently pass through the central axis of the superhelix, and the sites and entry and exit points of nucleosomes are more accessible because they lie towards the outside. So from where the DNA enters into the nucleosomes is more accessible because they are lying towards the outside, and in the central the axis of the superhelix is present. So these two are the proposed models for the organization of the 30 nanometer fiber, that how the nucleosomes present on the DNA is then bound on its own helix. Now both the models can be present, and in fact on the same DNA both the models can be present at different parts of the DNA, because this swirling around gives a negative helicity. In fact, swirling around the nucleosome in eukaryotic chromosomes give it a negative superhelicity to the entire DNA molecule. So this kind of things, the two models are present. And now then I discuss the positioning of the nucleosomes, which is the second part. How they are positioned is presence of other tightly bound proteins on the DNA. So there can be other kind of DNA protein molecules which can be present, which affect the positioning of the nucleosomes. So one positioning is because they prefer the AT rich in the minor groove, and they prefer the GC rich regions, GC rich minor grooves towards the outside. The second part is that the positioning of the nucleosomes is also affected by the other kind of proteins which is bound to the DNA. So sometimes what happens is that there are some other kind of sequence specific DNA binding proteins which are bound to the DNA. And when they are bound to the DNA, probably they leave a lesser amount of DNA in between, so a nucleosome cannot occur there, a nucleosome cannot be formed there. So this periodicity of the nucleosome arrangement after every 200 base pairs is irregularized in the 30 nanometer fibers. So as you can see, the sequence specific DNA binding proteins, when are bound, do not let the nucleosome to arise, or sometimes when they are bound, they bring closer the two nucleosomes which are supposedly be present at so much at the length of the DNA. So there are irregularities in the 30 nanometer fiber. So all we can say is that the chromatin fibers are more or less very flexible and dynamic and might interchange between the different folded states. The population of these states can depend on many factors, such as the presence of other chromosomal non-histone proteins, as we just saw, the modification state of core histones and linker histones, which is very, very important. The histone proteins can be modified at certain points, such as, for example, it can be hypoacetylated, it can be hyperacetylated or deacetylated, it can be methylated or demethylated. So histone core proteins, particularly certain amino acids such as lysine and arginine, can bear certain modifications, and because of these modifications the positioning of the nucleosomes is affected, which forms a very, very major point of regulation for the gene expression. So we will see when we will do the gene regulation that the modifications which are present at the histones or the linker histones affects the activity of a particular gene, or in fact whether a particular part of the genome is activated or not. Their modifications can play a role. The position of the linker histone on the nucleosome is also affecting the population of what kind of state is there, and then internucleosomal spacing. So as I told you, some non-histone proteins can be bound and therefore internucleosomal spacing can be large. Sometimes the internucleosomal spacing can be smaller because of the particular bias towards the AT rich minor groove binding and all those things. Therefore, the chromatin fibers are very flexible and dynamic and might interchange. Now, this packaging of DNA into nucleosomes, the 10 nanometer fiber and 30 nanometer fiber, results in the compaction of DNA length by approximately 40-fold. So, the DNA, which is a double helical DNA, is now 40 times compacted when it is formed in a 30 nanometer fiber. So, at the interphase, when the cell is not dividing and lying in the relaxed state, then at that point usually there is a composition of 10 nanometer and 30 nanometer genome, the chromatin fibers which are there. So that's how the genome is compacted inside the nucleus. So we further look at in a little detail about this mitotic or interphase chromosome, and now we go up the level or a higher order level structure from nucleosomal to a 30 nanometer fiber to now what is called as the 300 nanometer fiber or 700 nanometer wide fibers which are there. So we will see now how these kind of structures arise. These are termed as the higher order structure. The 30 nanometer fiber is organized into loops of up to 100 kbs that are held together at their bases by proteinaceous substance, which is the nuclear scaffold. Now, nuclear scaffold plays a very important role in giving higher order structure to the genome. The 30 nanometer fibers are now thrown into loops. So, around 100 kbp of these 30 nanometer fibers are thrown into loops and held together by the protein of the nucleus, or called as the nuclear scaffold protein. So, you can look at this, that is how the 30 nanometer fiber is thrown into loops, and therefore it gives rise to a chromatin fiber of the width which is 300 nanometers. And this organization is because it is interacting with the nuclear matrix containing the nuclear scaffold proteins. Two classes of proteins that contribute to the nuclear scaffold are topoisomerase II and SMC proteins or structural maintenance proteins. So, topoisomerase II, so you can see there is a 10 nanometer fiber, there is a 30 nanometer fiber, and then there is a further throwing into a loop. So, there is a central axis which is the nuclear matrix or the nuclear scaffold, and then this 30 nanometer fiber is thrown into DNA loops, which is then shown inside a box where you can see the 10 nanometer, and you might have a certain at certain times the unfolding of the loops into the naked DNA. So, the two kinds of proteins which play a role is the topoisomerase II and SMC proteins. So, we should recall I talked about the SMC protein in my last lecture, the structural maintenance proteins, which are basically the cohesins and the condensed. The cohesin keeps together the two sister chromatids, which is, for example, in prophase it is shown, and in metaphase the further two axes are basically the condensins also play a role and brings together the loops which are formed. The two sister chromatids are joined together by the cohesins, and then condensins further compact or bring together the loops which are there. At the anaphase, when the two sister chromatids separate, then the cohesin is basically is cleaved, and therefore the two sister chromatids can separate. But this kind of higher compaction is achieved by the structural maintenance proteins. Two major mechanisms by which chromatin is made more accessible. So, I would like to again emphasize on the fact that now the chromosome, the DNA is not lying open double helical structure, so that all the accessory proteins or all the proteins such as for transcription and replication can access it freely. It is wound around the nucleosome, and the nucleosomes are again into a 30 nanometer fiber kind of a structure, which is again thrown into a loop kind of a structure. So, the two major mechanisms by which chromatin is made more accessible are, one, that the histones can be enzymatically modified by the addition of acetyl, methyl or phosphate groups. So, as I talked about, histone modification is very important for all the kind of regulation or transcription or anything. The histone modification forms an important component, and then the histones can be displaced by chromatin remodeling complexes. Chromatin remodeling complexes are another set of proteins or enzymes which are present inside the cell, which can basically disassemble and assemble for the nucleosomal model. So, they can remodel the chromatin at different times, thereby exposing the underlying DNA sequences to polymerases and other enzymes which needs to work upon the DNA. So, these two are the major methods by which the chromatin is made more accessible despite their higher compaction and everything. So, a model for the structure of an interphase chromosome looks something like this. So, at interphase chromosome, you have got a 30 nanometer fiber which is thrown into further looped domains. So, the proteins, and then there is a nuclear scaffold which is formed, which is shown green in color, and that is how the DNA is present. When there are certain histone modifying enzymes are there or chromatin remodeling complexes are there, then these loops are unwound, the high level expression of genes in the loop. So, these loops are unbound, and then the expression machinery or the replication machinery can work on these decondensed loops which are present, and at these decondensed loops basically the DNA is exposed and the machinery can sit on the DNA in a sequence specific manner and do whatever function they need to do. So, interphase chromosomes are folded into a series of looped domains, each containing 20,000 to 100,000 nucleotide pairs of double helical DNA condensed into a 30 nanometer fiber. The individual loops can decondense when the cell requires direct access to the DNA packaged in these loops. The decondensation, as I already told, is brought about by the enzymes that directly modify the chromatin structure, as well as by the proteins such as chromatin remodeling complexes or RNA polymerases that act directly on the underlying DNA. The folded 30 nanometer fiber is anchored to the chromosome axis. The base of the chromosome loops is rich in DNA topoisomerases, one of an important protein for the maintenance of this looped structure, which are enzymes that allow DNA to swivel when anchored. So, when they are anchored, when they have to decondense, these DNA topoisomerase plays an important role, and structural maintenance proteins helps them or aids in forming a compact or a condensed state which is there. So, So that is how the interphase chromosome is basically at the molecular level. At the molecular level, the DNA double helical structure is thrown into such kind of 30 nanometer fibered loops lying at the relaxed state or an interphase stage. I would just review the features of the interphase chromosome which I started last time, talking about that when you observe it under a light microscopy, just a stained thing, you will see different kind of staining. Some parts of the nucleus will be highly stained or deeply stained or darkly stained, while some will be lightly stained. So there are two different kind of organization which we distinguish on the basis of this, which is called as the heterochromatin and the euchromatin. So I'll quickly go through this. The light microscope studies distinguish between two chromatin. As we talked about, the heterochromatin is the highly condensed form and the euchromatin is the less condensed form. The euchromatin is composed of the types of chromosomal structures, which is basically the 30 nanometer fibers and loop domains. However, in heterochromatin, there is a further more compaction which is there. So 30 nanometer fibers and the looped domains are the euchromatin region, which is mostly the active part, and the heterochromatin part is a further kind of compaction and conduction. Condensation occurs in the heterochromatic region, which is the darkly stained. So we discussed the euchromatin versus heterochromatin part. The most important part is that the euchromatin is genetically active, whereas the heterochromatin is said to be genetically inactive and late replicating. However, this is not completely true because some of the genes are also located in the heterochromatic part and may be transcribed when needed. But most of the heterochromatic regions are non-transcribing and late replicating regions. So we talked about this euchromatin and heterochromatin. Then we also talked about two types of heterochromatin, which is the constitutive and the facultative heterochromatin. The facultative heterochromatin, which is not heterochromatinized always. It is not always found in a heterochromatinized state and can transform between the heterochromatin and the euchromatin. For example, we talked about the X chromosome, which is condensed in a female cell, but when it is passed on to the male cell in the next progeny, it is decondensed, and if it is passed on to the female in the next progeny, then it can be decondensed or condensed. So it is a facultative heterochromatin. The constitutive heterochromatin are the permanent features of a chromosome, as we will see the centromeres and telomeres, they are heterochromized part. The constitutive heterochromatin is always compact and tends to be enriched in repetitive, gene poor and late replicating DNA sequences, whereas facultative heterochromatin can reversibly undergo transition. So we talked about the difference between the constitutive. The properties of heterochromatin is therefore that the heterochromatin is highly condensed, it is late replicating, it is methylated. So the modifications are there, many kind of modifications are there in the heterochromatic DNA. It is methylated. In heterochromatin, the histones are also modified, they are hypoacetylated, usually at lysine amino acid residues. Then histones from the heterochromatin are also methylated on lysine 9 position. It is transcriptionally inactive more or less, and it does not participate in genetic recombination. So no crossing over occurs in the heterochromatin part, and it has a gregarious instinct. That is, many of the heterochromatins tends to associate together, as there is in the polytene I just mentioned the polytene chromosomes in the dipteran larvae, where the heterochromatin centromere basically joins together to give rise to a structure, and different DNA fragments are then protruding. So it has got a kind of an aggregating instinct which is present. In lower eukaryotes, nearly the entire genome is in an open conformation, and only regions important for genome integrity such as telomeres and centromeres are kept stably heterochromatinized and referred to as the CHC. However, in higher eukaryotes, the same chromosomal regions are also compact. So centromere and telomeres are also heterochromatinized in higher eukaryotes. However, due to the enormous increase in genome size and organismal complexity, a large percentage of the genome, including the repetitive and the non-coding sequences, is wrapped into CHC. And that's what, when you observe the interphase nucleus, you see different portions as heterochromatinized and euchromatinized. So heterochromatinized portions contain the centromere as well as the future telomeres of the condensed chromosomes, but they are also containing certain very highly repetitive sequences which are present in the case of eukaryotic chromosomes. So these are also constituted constitutive heterochromatin, the transcription at these genomic regions is possible. As I said, that it's not necessarily that the heterochromatin parts are wholly non-transcribing or non-active, but the transcription at these genomic regions is also possible, and in certain instances even required for the maintenance of these constitutive heterochromatin region. However, the transcript levels are low and they do not match that of the protein coding genes in the euchromatic environments, and therefore in comparison to the euchromatin, we say that the heterochromatin is a non-transcribing part of the genome. Similar to CHC, which is the constitutive one, the facultative heterochromatin is also transcriptionally silent but retains the potential to interconvert between the heterochromatin and the euchromatin. So, the facultative heterochromatin can be molecularly defined as condensed, transcriptionally silent chromatin region that decondense and allow transcription within following contexts, whether it is a temporal difference, for example developmental states or specific cell cycle stages. So, a particular part of the DNA which remains heterochromatin at say gap phases or G1 or G2 phases become transcriptionally active in some other phase of the cell cycle, then it is said to be facultative heterochromatin. Spatial, for example nuclear localization changes from the center to the periphery or vice versa due to exogenous factors or signals. So, if a certain portion is present in the nucleus center, then it might be euchromatinized, but however when it moves towards the periphery it becomes heterochromatinized. So, it is a facultative heterochromatin or parental or heritable, for example the X chromosome. So, if it is present in a female, one of the X chromosome remains condensed, but if the same X chromosome is passed on to the male in the next progeny, then it will become decondensed or a euchromatin in origin. So, the facultative heterochromatin is distinguished by the differences in the temporal, spatial or parental or heritable context we talk about the facultative heterochromatin region. Now, what happens, how this FHC can be established? The FHC establishment and or maintenance is achieved by a combination of processes. So, the combination of the processes which are there is the incorporation of specific or alternate chromatin components. So, as you can see, there is a facultative heterochromatin, it can change to form the euchromatin, and the factors which are responsible for them can be the certain kind of chromatin components. That association of certain chromatin components can convert the euchromatin into a facultative heterochromatin. For example, linker histone H1, the modifications at histone proteins or histone variants are sometimes present which can convert the euchromatin into facultative, or as I said, the modification, the methylation, the deacetylation or the hyperacetylation of the histone proteins. Then the chromatin modulation, the chromatin remodeling complexes can also convert the euchromatin into a facultative heterochromatin and vice versa. And then there are concerned action of certain transactin factors such as the non-coding RNAs or the transacting proteins. For example, certain proteins are given which when associated with the facultative heterochromatin can convert it into euchromatin or vice versa. And then the last thing was the subnuclear localization, as we talked about the spatial context which leads to the formation of the facultative. So, all these components can basically transform the euchromatin into facultative heterochromatin or the vice versa thing. So, at the interphase chromosomes, we see that there is a whole chromatin which is present, which will later on give rise to the chromosome. So, we know that they condense and form the highly condensed chromosomes which are there present in the metaphase chromosomes. So, one important aspect that I want to talk about here is that the arrangement of the chromosomes. So, the chromatin is basically like a bowl of noodles. So, the bowl of spaghetti which are tangled around each other. So, is this tangling up random? That when the highly condensation part will occur, so all these chromosome number 1, 2, 3, we know that we have 23 pairs of chromosomes. So, all these 23 pair of chromosomes which will be formed in metaphase, then all these 23 pairs, the 23 pairs of 23 chromosomes which are present, are they just lying tangled together inside the interphase nuclei? So, another aspect when we look at the interphase nuclei is that how they are arranged. So, the chromosomes occupy specific regions of a nucleus. So, these spaghetti is not tangled randomly. It's not like one loop and another loop is all tangled around in the interphase nucleus, and later on they somehow get detangled and become the chromosome number 1, 2, 3, 4. It is not. At the interface also, the chromatin fibers, they occupy specific regions which are called as the chromosome territories. So this figure explains it better, that if you can see, then this is a stained diploid spread of chicken metaphase chromosomes. The same spread after multicolored fluorescent in situ hybridization with pseudo-colored chromosomes. So all what I am trying to say is, if you see at metaphase, then you can see two pair of chromosomes of a single color. So chromosome 1, 2, 3, 4 are marked by these colors. For example, chromosome 1 is shown red. Now if you stain with the same probe at the interphase nucleus, then still you can see that there is a territory which is defining the part of the interphase chromosome which will become chromosome number 1. Although the sister chromatids are at separate regions which are present, which will together come and form a pair, but when you see, the probes were detected when the mid-plane light micro-optical section through a chicken nucleus shows mutually exclusive chromosomal territories with homologous chromosomes seen in separate locations. So the homologous chromosomes will come together at a later stage, but at the interphase nucleus, the tangled mass of chromatin is actually localized into chromosomal territories, and these chromosomal territories represent basically the future chromosomes which will be there. So they are not just randomly arranged. As you can again see in this diagram, that there is an X chromosome which is there shown by an arrow, and in the interphase nucleus you can see the localization of this X chromosome which is present. So the chromosomes are arranged in a particular manner. So we will come to the last part, which is the metaphase chromosome, which is the highly compacted form. It looks like this, the highly compacted form chromosome structure, and it has certain special features, which is the presence of a centromere, presence of a telomere. So there is a nucleus. So every part of the, when it is zoomed, there is a nuclear matrix, and then there are loops of 30 nanometer fibers which are. So further higher condensation which occurs in here. So there is a primary constriction, there is a secondary constriction. The part of the chromosome after the secondary constriction is termed as satellite, then the end are termed as telomeres, then there is a kinetochore. We will talk about all these things, and different parts of the chromosome can be heterochromatinized and euchromatinized. So the centromere was first described by Walter Fleming. It is not always found in the center of the chromosomes. If it is in the center, then the chromosome is termed metacentric. If it is towards one pole, it is submetacentric. If it is further towards one of the end, it is acrocentric. And if it is present at the end and there are only two arms of the chromosome, then it is said to be a telocentric chromosome. The eukaryotic chromosomes have a single centromere that ensures their segregation during mitosis, because basically the spindle fibers form around the centromere. There is a protein structure which is called as the kinetochore, and the spindles are arranged, and then the two sister chromatids do separate. So they contain repetitive DNA and modified nucleosomes. They are the part of the heterochromatin region. The centromere proteins CENP-A and CENP-B are inner centromere proteins that interact directly with the DNA. CENP-A is an unusual variant of histone protein, histone H3, that replaces H3 in the nucleosome. So peculiarity at the centromere found exclusively at the centromere, and they CENP-B as a protein that binds the simple sequence repeated many a times in the centromeric DNA. In humans, these repeats are 1 base pair in length, and it is called as the alphoid DNA. So 1500 to 30,000 copies of alphoid DNA per chromosome are present, and they span a region of between 0.9 to 5.2 MBs. The centromere is a region of specialized chromatin found within each constricted chromosome, and that is called as the primary constriction, and that provides the foundation of the kinetochore assembly. So what is a kinetochore? The primary function of the centromere is to provide the foundation for assembly of the kinetochore. Kinetochore is a protein complex essential to proper chromosomal segregation during the mitosis. So kinetochore are the protein molecules where the spindle fibers attach and then constricts to move the two sister chromatids towards two different poles. So the mature kinetochore is a complex of about 80 or more proteins. Its functions involve microtubules, as I said. The other proteins are part of the spindle checkpoint that allows only cells with correctly assembled spindles to pass. So, there are checkpoints in the cell cycle. If there is a correct organization of the spindle fiber, then only it can move to the next phase. The centromere identity is preserved during cell division. However, the kinetochores are dynamic assembly. So, they form and they dissociate, but centromeres are constitutive heterochromatin, they are maintained. Centromere serves as a site for sister chromatid attachment. The chromatid is one half of two identical copies of a replicated chromosome. Sister chromatids originate from single chromosome, while non-sister chromatids, just to differentiate between the two. So, chromatids in the early stage of the these are some of the terms which are used. The chromonemata, the chromatids in the early stage of condensation are called chromonema. Chromomeres, they are serially aligned small bead-like accumulation of chromatin material that are visible along length of chromosome, especially during the zygotine stage of meiosis. So, that is how the chromosome, metaphase chromosome looks like. The end of the metaphase chromosomes is termed as telomere. The telomere protects the ends of the DNA molecule contained within the chromosome from attack by the exonuclease enzymes or the repair enzyme. They contain special sequences which are the repetitive sequences such as TTA-GGG in humans, the GC-rich strand which contributes to the 3' end of the DNA molecule. The nucleosomes are not present in the telomeric region of chromosomes. So, it is a naked DNA, it is free of nucleosomes, and two special proteins are associated and bind to the repeat sequences, the TRF1 and TRF2. The eukaryotic chromosomes bend back on themselves because it is a repeated sequence, so it kind of form the bend over itself called as the T-loop. The formation of the T-loop requires both telomeric repeats and the single-stranded G-tail. So, when you have done replication, you must have done that the telomeres, because they are single-stranded at the end of the present. So, how the loose ends are basically replicated. So, this is the proposed structure. And telomeres tend to shorten in cells without telomerase over time, and cells may stop dividing and undergo senescence. And to prevent this, basically there is a special kind of replication which occurs. So, telomerase is the enzyme which binds, and because of this T-loop structure, it tends to help the single strand which is formed after the replication to replicate. So, that is telomerase. And the last is the secondary constriction, which is again present in the metaphase chromosomes. So, one or more secondary constrictions can be found. The secondary constriction is called as the nucleolar organizer region, which is necessary for the formation of the nucleolus. It appears at one end of the chromosome part beyond the called as the satellite. So, as I told you, there is a chromosome chromatid which is there, then there is a primary constriction which is the centromere, and there is a secondary constriction. The part of the chromosome after the secondary constriction till telomere is said to be the satellite. It is not to be confused with the satellite DNA. Satellite DNA is another term which is used for the repetitive sequences which are present in the DNA molecule. So, there are certain repetitive, highly big repetitive sequences, more than 100 base pair sequences which are repeated all over in the eukaryotic genome. That is termed as the satellite DNA. But satellite is that part of the chromosome which is present beyond the secondary constriction in the metaphase chromosome. Now, what is the role of this secondary constriction? Around this secondary constriction, the nucleolus is organized. So, nucleolus of the cell, that is why it is also termed as the nucleolus organizer region. So, nucleolar organizer region is necessary for the formation of the nucleolus. It appears at one end of the chromosome part, beyond that is called as the satellite. The chromosome is called as the SAT chromosome. So, there is a SAT DNA and there is a SAT chromosome. SAT DNA is the repetitive DNA sequence, whereas SAT chromosome is the part of the DNA which is present in the satellite part of the DNA. The human chromosome 13, 14, 15, 21 and 22 in NOR contains several hundred copies of genes. So, these SAT chromosomes are human chromosomes 13, 14, 15, 21 and 22 which all contain secondary constriction, and these NOR contain several hundred copies of gene coding for ribosomal RNA. So, repeated sequences, repeated genes are present which code in for the ribosomal RNA and therefore helps in the organization of the nucleolus. Sometimes there is another secondary constriction also present which is termed as secondary constriction 2. The location is constant for a particular chromosome. Long arm of human chromosome number 1, 10, 13, 16 and Y chromosome are said to be secondary constriction 2 which is present, and the location is constant for a particular chromosome. For secondary constriction 1, it can be the location is not fixed also, although it is on one of the arms, or it is present on one side of the arm of the chromosome which is there, but the position is not fixed. Secondary constriction 2, the position is fixed for a particular chromosome, and the long arm of human chromosome 1, 10, 13, 16 and Y contains this secondary constriction 2. So what we discussed today is basically the eukaryotic genome organization, in which we talked about the nucleosomal organization. So we looked at the different levels. The first was the DNA double helix wound around the nucleosomal, giving rise to a 10 nanometer fiber structure. Then further up, this 10 nanometer fiber chromatin is wound around itself on its own axis, giving rise to the 30 nanometer fiber structure. This 30 nanometer fiber is thrown into looped domains to give rise to the interphase look of a chromatin, and in that chromatin we can distinguish between the euchromatin and the heterochromatin part. The euchromatin part is basically this 10 nanometer, 30 nanometer thrown into loop domains which is there, and heterochromatin is further highly condensed. Then we also talked about the two types of heterochromatin, which is the constitutive and the facultative heterochromatin, and further going higher order in the structure, we talked about the metaphase chromosomes, which is the most condensed state of a genome in the eukaryotic system, and this compaction is attained by molecules and enzymes such as topoisomerase 2, the nuclear scaffold proteins, as well as the structural maintenance proteins which are there. And in this metaphase chromosome, then we talked about the certain features such as the presence of centromere, telomere, kinetochore and secondary constrictions, which are important parts of the metaphase chromosome. So next time we will talk about the further up, that how DNA transcribes into the RNA. So for now, that's all.
I would like to thank ma'am for this interesting discussion. Thank you, ma'am. And thank you, dear friends, for watching. Stay tuned and keep watching. Thank you.